Hello, everyone, and welcome to the Brain Matters webinar series of the Human Brain Project. Entering its third and final phase, the Human Brain Project has launched Brain Matters, a webinar series which explores scientific issues being tackled by the HBP scientific community. These hour-long sessions focus on different areas of brain research and feature expert speakers with the goal of highlighting the HBP's scientific achievements and the state-of-the-art services offered by its infrastructure for brain research, EBRAINS. My name is Hans Ekkehard Plesser, and I will moderate this 15th edition of Brain Matters on connectivity concepts in neuronal network modeling. This webinar features the Human Brain Project researchers Johanna, Senk, Mikael, Djurfeld, and Sacha from Albada, as well as Birgit, Kriener. Thank you for taking your time to join us today. During the webinar, each speaker will give a short presentation of approximately 10 minutes. Feel free to write your questions using the Q&A function in your Zoom app during the talks. We will share and discuss your questions at the end of the webinar. When you ask questions, please mention the name of the speaker you would like to address with your question. Please also note that we will not read the chat in Zoom. We will only read the Q&A section. Oops, sorry, there. So a little bit more about me. I'm a professor at the Norwegian University of Life Sciences and also visiting researcher at Forschungszentrum Jülich. And as you can see, I recently shortened my beard a bit. I'm a lead developer of the NEST simulator, and I lead the high-level support team of the Human Brain Project. And I will be the moderator for this webinar. Connectivity concepts is the topic of today's webinar, and if you look just at nerve cells, they can't do much if they are not connected. Now, if you create a model of the brain and tell brain researcher, okay, here are 4 nerve cells, connect at random each nerve cell with 2 other nerve cells, then one researcher might go about it like this. So from each nerve cell, 2 outgoing connections to other nerve cells. But another researcher might understand the instructions differently and have 2 incoming connections to every nerve cell. Now, we look at these figures, they look pretty similar. So does it make a difference? And it does indeed make a difference if we create a slightly larger network and let that run, look at the activity in this network, then we can see that no matter which of the 2 connection options we choose, the overall activity looks relatively similar. But if you look at the right here at the distribution of activity in individual nerve cells, we actually see quite a difference, a lot of variation here, and very uniform activity profiles here. So details in connectivity make a difference, but we often find in scientific publications descriptions in textual form, such as connect each neuron with 10% of the other neurons at random. And to look at this in more detail, the speakers of today's webinar, together with some more colleagues, recently published a paper where we reviewed this in detail. And in this webinar, we will go through the results of this paper, our key findings. We will start with Johanna Senk, who will summarize how neuronal networks are used and described in the computational neuroscience community. Johanna is a postdoctoral researcher at the Forschungszentrum Jülich in Germany. Welcome, Johanna. I'm looking forward to your presentation.
Thank you very much. Let's first get an overview of the networks used in the computational neuroscience community. We reviewed network models for which both article and code were published to find out which connectivity structures do computational neuroscientists use in their models, how do they define which neurons of a source population connect to which ones in a target population, and in particular, which terminology do they use to describe these models. Focusing on network connectivity, we review network models of point neurons, simple multi-compartment neurons without considering connectivity specific to compartments, and neural mass models, but exclude neural field models as well as more detailed ones. We account for connections between neuron populations and in that sense exclude connections with external stimulating and recording devices. The neuron dynamics can be of type spiking, binary, or rate. We do not a priori exclude plastic networks in which the connection strengths or even the connections themselves change over time, but we only consider structures that are independent of dynamics, meaning the result of an initial network construction. The network models included in our study are in the repositories ModelDB and open source Brain. Both repositories have been maintained for many years, or in the case of ModelDB, even decades. They provide different ways to curate, further develop, visualize, and even simulate models. ModelDB uses the infrastructure of SenseLab of Yale University and mostly aims to serve as static reference for published articles. No restrictions on programming languages or simulators are made. To match our scope, we narrow the broad collection down to the section MicroCircuitDB, Connectionist Network. On the contrary, open source Brain stores models in public version-controlled repositories such as GitHub to foster ongoing and collaborative model development. Here, models are standardized and made available in the model description languages Neuro, ML, or Pine, besides potentially further versions. We analyzed a total of 42 network models found in these repositories that are in line with our scope, and we characterized them according to certain criteria: the metadata, the description of connectivity, the implementation of connectivity, further network specifications, and connectivity concepts. On the following slides, I will show the results of this literature review. As metadata, we consider when, where, and by whom were article and code published. About 80% of the selected models are indexed by ModelDB and about 20% by open source Brain. You can see that the articles we looked at appeared in a number of different journals. They are mostly from the recent years and were mostly developed through collaborations of a small number of different labs, mainly from Europe and North America. These observations reflect that scientists increasingly appreciate the value of reproducible research, which leads to more published code and more use of dedicated repositories. Journal policies also play a role. As an example, the Journal of Neuroscience, which is here overrepresented, explicitly recommends to deposit model code in repositories such as ModelDB. How does the article describe the connectivity, and is the description complete? For instance, the location of the description. Mostly, at least a large part of the description was found in the main manuscript, but combinations with a part in a supplement of the publication or part in an external publication referred to were also seen. Reasons for incomplete descriptions in the main manuscript could be space restrictions by the journal, or in case that a model builds on previously published one, the authors decided to describe only the differences. Connectivity was described using text, often together with illustrations, equations, and tables. We further ask if the code used for the implementation is mentioned in the article. A possible explanation for the large fraction of no code specified is that the code was published in the repositories after the article was out. In case of probabilistic connections, we identified a non-negligible number of articles that described the connectivity as random or gave just a connection probability without further specifications. In total, we classified 15 out of the 42 articles as ambiguous with respect to how they describe the connectivity, meaning that details were imprecise or completely missing. Moving now from the article to the code, how is the connectivity technically implemented? General purpose software such as MATLAB, Python, C or C++ was used, or dedicated neural network simulation software such as Neuron, Nest or Brian. For the implementation of the connectivity, we distinguish between custom and built-in code. Built-in here refers to standardized routines provided by a simulator. For example, a high-level function like nest.connect. Surprisingly, the large part of the studies we looked at used pure custom code, meaning they wrote the connectivity from scratch. How are network nodes and edges characterized? To better understand the studied networks, we ask what the connected units or nodes represented, single neurons or groups of neurons, and by which dynamics they are described, rate spiking or binary. As mentioned before, for plastic networks, we only considered the initial connectivity for evaluating the connectivity concepts used. Finally, we bring article and code together and ask which connectivity concepts are realized in the studies. Are deterministic and probabilistic connections present in the network? Have the connections a spatial component, for instance, a distance dependency? The most common deterministic rule we found was all-to-all. And the most common probabilistic rule, pairwise Bernoulli, in which a connection probability determines if a connection between two nodes is established or not. And we specify self-connections, autopsies, and multiple connections between the same pair of neurons, multopsies, can occur. Although these connectivity concepts appear to be quite basic and fundamental, we classified more than a third of the studied descriptions of connectivity as ambiguous. We assume that one reason for that is a lack of readily applicable standardizations, tools, and references, which we addressed here in this study. With this, I'm at the end of my part. Would like to thank you and stop here.
Okay, thank you very much, Johanna. So thank you very much for keeping time very well. And with that, we move on to Mikael Djurfeld, who is a researcher in neuroinformatics at the KTH Royal Institute of Technology in Sweden. He will talk about how connectivity concepts are expressed in programming languages, are created in simulation software. Varsågod, Mikael.
Thanks. Most simulators typically provide an idiosyncratic model description language. For example, Nest and Neuron have their own primitives for building a model. But there are also cross-simulator description languages. For example, Pine, NeuroML, 9ML, CSA, and Sonata. The advantage of these are that it makes it possible to move a model between simulation environments. For example, some Pine models can be run both in Nest and Neuron, then facilitating, for example, model calibration. This also facilitates sharing and publication. There are different types of descriptions of connectivity in these languages. There are procedural descriptions where you specify an algorithm for setting up individual connections. A different kind of description are the declarative ones, where you don't specify the details of the stepwise setting up of connectivity. For example, here you have a source and target population and specify a rule for setting it up. The advantage of declarative descriptions is that you give headroom for the simulator to optimize how connectivity is set up. This is especially useful if you have a parallelized simulator. One special case of declarative descriptions is the algebraic description of the connection set algebra, and we'll talk a little bit about that later. So when surveying the model description languages used, we found that there is a set of connectivity rules which is present in many of the simulation environments. So here on the left-hand side, you see a set of connectivity rules. So we have the one-to-one connectivity, all-to-all, for example, and the pairwise Bernoulli connectivity and a set of other random connectivity types. And as you can see, most are supported by many of the languages in the columns, Nest, Pine, NetPine, and 9ML, with small variations, like if they support setting up connectivity with or without autopsies or with or without multipsies. Now, a few words about the connection set algebra. This is a declarative algebraic description of connectivity. In CSA, neurons are enumerated using integers. Because of this, a connection can be represented by a pair of indices, the source and target neuron. The connection set algebra works with sets. So you have sets of such connections, sets of pairs of indices, and there is an algebra on these sets whereby you can specify new sets of connections. As an example, omega here is a set of all possible connections. Notice that this is an infinite set. The advantage of being able to work with infinite sets is that you abstract from and generalize from networks of a specific size. Delta is a set of all autopsies, or another way to view it is one-to-one connectivity. So the meaning of this is that neuron 0 connects to neuron 0, neuron 1 connects to neuron 1, and so forth. So using these two connection sets, you can express all-to-all connectivity without autopsies as an algebraic expression using set difference. When you apply this to a specific network of finite sites, you can use the set of all possible connection in that network, X, and create an intersection with the infinite sets. In this way, CSA can capture the essential character of a type of connectivity through abstraction. Thank you.
Thank you, Mikael. Let's see, so Sacha is a junior professor at the University of Cologne and leader of the research group Theoretical Neuroanatomy at Forschungszentrum Jülich in Germany. She will describe how one can create unambiguous network diagrams to graphically present network models.
Thank you. Neural network models are often not just described mathematically, but also illustrated with network diagrams. And therefore, we have also tried to come up with a consistent and unambiguous graphical notation for neural network models. My presentation is not really cooperating somehow. Okay, so to give an example of the different notations that are in use, excitatory and inhibitory connections are often depicted in different colors that in one notation one might refer to as the water tap notation here on the left, where the excitatory connections are given in red and the inhibitory ones in blue. And on the right, the so-called, or what one might call bank account notation, where the inhibitory connections are in red instead, and the excitatory ones in black, like here, or sometimes also in blue. Also, you see that the arrowheads are different. So on the left side, both excitatory and inhibitory synapses are indicated with little balls at the end, and on the right, the inhibitory connections are indicated with these discs, and excitatory ones with triangles. So in our graphical notation, we did not focus on color because that's a whole other ball game, let's say, and we rather focused on the shades, and this is the notation that we came up with. So the nodes themselves, they might represent single units, or they might represent populations of cells, and we proposed using thicker edges, or edges maybe not so a good word when relating to graphs, but here so the thicker circumference, let's say, of the nodes to depict a population compared to a single unit. And then excitatory cells, because often triangles are already used because of pyramidal cells. So following that intuition, we stick to a triangle, and then we have circles for inhibitory nodes. If you want to put a bigger label inside, you might turn it into an ellipse, then it's still distinguishable from the other types of nodes. Here we also took into account stimulating and recording devices. And then beyond the nodes, also the connections between them, of course, are important. That's our main focus. One might depict deterministic connections with solid lines connecting the nodes, probabilistic connections with dashed lines. And then the arrowheads, we chose them, or we proposed them to match the symbols for the excitatory and inhibitory nodes for consistency. And then these edges, they may be annotated with the symbols that that Birgit was going to introduce for the different types of connectivity concepts, with one-to-one with a delta, all-to-all connectivity with an omega, an explicit set of connections with an X, pairwise Bernoulli connectivity with a P, and so forth. It's details that are often left out of neural network descriptions are whether or not autopsies, so self-connections, are allowed, and also whether or not multapses are allowed, so multiple connections, directed connections between a given source node and a given target node. And so we propose indicating that explicitly. Then parameters can be indicated, where we propose an overhead line for a constant parameter. And if a parameter is distributed, so for instance, the weights or delays are distributed, then one might also indicate that the distribution type, at least. It may become too cluttered if you also give all the values of the parameters. Those might be given in different places in the document. But in any case, this already would reduce the ambiguity a lot of many published models. And I give a a few examples. One is the classical balanced random network model of Brunel. And it consists of an excitatory and an inhibitory population of neurons that are recurrently connected and also within each population, with probabilistic connections here indicated with the dashed lines. The in degrees are fixed, so that refers to what Hans Ekkehard showed. So that's an important detail to know, that the network would behave differently if the out degrees, for instance, instead of the in degrees were fixed. Multapses are allowed and odd tapses are not allowed. And the external drive is connected. Here we denote it with a one-to-one connection because all the neurons receive an independent Poisson input. Then such diagrams, they may be complemented in manuscripts with different types of descriptions of the connectivity and the network as a whole. One may be just in text. Tables can be useful. One can use also the consistent mathematical notation of the connection set algebra that Mikael introduced. Here, this row 1 refers to connectivity with fixed in degree. And then the M indicates that multapses are allowed. And this is the set of nodes, source and target combinations that the connections are established on. Then, as Mikael also showed, high-level connectivity concepts are also implemented in various simulators. Here we give an example from the NEST simulator, how one would define the connectivity of this balanced random network model. Another example is more involved. So it's a cortical microcircuit model distinguishing layers 2, 3, 4, 5, and 6. And within these layers, there are different populations of inhibitory cells, somatostatin expressing interneurons, vasoactive intestinal peptide expressing interneurons, and parvalbumin interneurons. And we showed this also to demonstrate or to illustrate how our notation is intended to create unambiguous diagrams, but they may not be the most intuitive to look at. So you can still keep complementing your diagrams with more intuitive ways of depicting the connectivity that are maybe that focus on different aspects of the connectivity that can also use color, etc. Here, the connections are also probabilistic. This is indicating what the connectivity is like qualitatively within each layer, using an illustrative inhibitory population. And here the weights and delays are drawn from log normal distributions. And there's an external thalamic drive that is also connected probabilistically with the populations. So far, we have considered neurons that are not explicitly located in space, but of course, neurons do have a location in the brain, and they have distance-dependent connectivity, and they have patchy connectivity, as illustrated here from anterograde tracing in New World monkeys in layer 3 of cortex, and then you see, especially in layers 2 and 3, that these connections they terminate in spatially more distant patches. And then the local connectivity, so the intensity of the staining here, it decays exponentially with the distance. Here 2D profile was fitted to the layer 5 connectivity. To capture also such spatial connectivity in diagrams, Birgit Kriener and Nicole Voges came up with an example also of local and patchy connectivity. What is maybe especially useful about this example is that it makes explicit what are all the aspects of such a network that you need to specify in order to have a complete description. That some of these are sometimes forgotten, that also the boundary conditions play a role, and that the metric should be specified. And then, of course, the spatial profiles of the connectivity. So that already brings me to the end. Besides the people presenting here, a few more authors were involved in the study that was published in PLOS Computational Biology. And hereby I would like to thank them. And in addition, the people listed at the bottom have provided useful inputs for which we are grateful. And with that, back to Hans Ekkehard.
So let me introduce Birgit again. She is a researcher at the Center for Molecular Medicine in Norway and has long experience in computational neuroscience, particularly focusing on network dynamics. Today, she works in data science and deep learning to develop diagnostic tools for pediatric brain cancers.
Yes, thank you. Sorry about the technical difficulties. So I wanted to talk to you about connectivity concepts. So we already heard a little bit about it. But if you think of connectivity, then it's important to have a notion of what it means. So, and usually we think about it in terms of graphs. So a graph consists of n nodes, where the nodes could be entities like neurons or websites or whatever you like. And these nodes are somehow connected by edges. And these edges for neurons could be synapses, for websites could be links, and so on and so forth. So it's a very abstract concept. It's just an abstraction of a network reduced to who is connected to whom, or that's also sometimes called network topology. A very nice concept to quantify a graph is you just arbitrarily assign names to the nodes, and then you just make a lookup table. So who's connected to who? For example, there's node 1, it's connected to node 2, and node 2 is also connected to node 1. So you make a 1 at 1 goes to 2, and 2 goes to 1. And then there's a third node, which might be connected to 1 and 2, and so on and so forth. So you fill this lookup table up, and then you have a nice representation of your graph or your connectivity. So if you want to specify connectivity, that is actually the most precise and reproducible way to specify it. But it's a little bit of an overkill if you, for example, have a very, very large networks, and especially if the connectivity can be expressed by simple deterministic patterns such as all-to-all or one-to-one. And another case where it might be overkill is if you have probabilistic models whose results should not depend on individual realizations. So you might not be interested in one concrete instantiation, but more on a general concept of connectivity. And then it's often maybe easier and more practical to specify concise, unambiguous rules. So connectivity comes in different flavors. Local connectivity, we already saw that in Sacha's presentation, where you, for example, ignore space and draw from all nodes in a probabilistic manner. But there also might be spatially derived connectivity, where connection probability varies with distance. And of course, you can have even more complex connectivity within and between layers or neuron types and so on and so forth. So where you would have another rule maybe to connect these. And often commonly reoccurring patterns can be identified. So the idea is to derive connectivity concepts which kind of specify these patterns. And these concepts then induce different graph theoretical characteristics, such as the degree distribution, which can be helpful then to specify the network and to make it testable even further. So what is a degree distribution? It's actually a very simple concept. So you just go through the nodes and count how many connections they have. So for example, this red node has 4 connections, this blue node only has 1, the orange one has 2, this one has 5, this one has 4 again, and so on and so forth. And if you do it for a while, you get a nice histogram, and this is what we call the degree distribution. If you have a directed network, like for example, between neurons, often the case, right? then the adjacency matrix is typically asymmetric, and the in degrees and out degrees don't have to be the same, right? They can have different distributions. So what does this mean concretely now? So for example, if we have a set of source nodes and a set of target nodes, which of course can be identical, so then we already heard about some of these rules, 1-to-1. That would mean each node in S is connected uniquely to one node in T. All-to-all means simply that each node in S is connected to all the nodes in T. For example, here, every of the 5 nodes connected to all of the 5 nodes in the target layer. And the degree distributions in these cases are very simple, right? So for A, we have a degree distribution of 1. Everybody has an out and in degree of 1. And in B, everybody has an out degree and in degree of 5, or if it's an n-dimensional network of n. A little bit more complicated does it get if you, for example, have a probabilistic rule, for example, pairwise Bernoulli. That means that each pair of nodes with source in S and target in T is connected with some probability P. So since you roll a die with probability, for example, 0.5 in this example, for each of the connections, you get a binomial degree distribution. So this is what you would expect. And this is also what you can test, of course, if the network really fulfills this. If you, for example, have random connectivity with a fixed total number of connections, but without multiplexes, as in this example, then it's a little bit more complicated because once you already had one connection drawn, you cannot draw it again. So that means you cannot choose that node anymore. So what then typically happens is that you get some kind of hypergeometric distribution, or in this case, a joint distribution of the network would be a multivariate hypergeometric distribution because you have these categories. So for all the individual nodes, right, it depends on each other. Another example would be, for example, random fixed total number with multiplexes. So in this setting, you have your source and target groups, and then you just throw in k-sin connections at random from all the possible connections. And you can choose connections several times, like, for example, here in this example, where S0 is connected twice to T1. Sorry for I didn't fix in degree distribution here. I'm sorry about that. In this case, you get a multinomial distribution. So what does this help us? Well, connectivity concepts, they allow an unambiguous network specification. It can be expressed in various formats. Most of them we already heard about, like a rule, the connection set algebra, a table, or a graphical notation. The in and out degree distributions can often be inferred. And that gives us useful dynamical information about the spread in the input and output. And most of all, what is also useful if you want to test your simulator, if it actually creates the right networks that you think it creates, you can use statistical tests to, for example, check if the degree distribution is really the one you expect. So if I have 2 more seconds, or 1 minute, of course, you can also try to come up with concepts for spatial networks. So spatial networks, there you have nodes embedded into some metric space with a certain node density rho here. So it could look like this. And then you just have specify a connection profile, means how many nodes do I see in my vicinity. Then you establish connections according to this connectivity profile. And that means this way you build up a certain network. And to specify these spatially embedded networks, you would have to specify the metric. So what defines the distance in that space? The dimension of the network, commonly 2 or 3, but you can also have 1-dimensional ring networks, for example. You need to specify the boundary conditions. Are they open or periodic? Then, of course, a node distribution. It could be just randomly distributed, or the nodes could be sitting on a grid. And the connection profile. So commonly profiles would be just a direct dependence on the distance, or an exponential decay, or a Gaussian profile. So there are several choices. Okay, and then, Then, yeah, the degree distribution can also be inferred, but it's usually quite complicated. And with this, I'm finishing. Thank you.
Okay, thank you very much, Birgit, and thanks again to all the speakers. We are now ready to engage in the question and answer session. I see that a few questions have already come in the Q&A. And if you would like to ask more questions, please use the Q&A feature at the bottom of your Zoom window as shown in the slide now. And then I would invite the speakers also to turn their cameras on again now for the Q&A. And the first question is to Johanna Senk. So what might be a reason why so many studies used custom code for defining the connectivity while simulators already provide high-level functions for this purpose?
Yeah, thank you. This is a really good question. And I think it goes, the answer, there are several answers to this, and one could go in the direction of why dedicated simulators are probably not so widely used as we might wish, and that could have one reason could be that the researchers come from a lab that, for instance, traditionally uses MATLAB, and so the researcher directly starts to use this for the implementation. Another reason could also be that there is, of course, an entry barrier. It is difficult to learn a simulator language if one is new to it, and people rather would like to have full control over every detail. But yeah, we would like to emphasize here that it's worth it to get into it, because then one can rely on well-tested code that is also efficient and more readable if one uses these high-level connectivity functions that are more concise than, let's say, multiple loops for representing an adjacency matrix in a general purpose language. Yeah, so I think this is something that will hopefully change in the future. In particular, when these high-level connectivity functions in the simulators can be more linked to such concepts, these mathematical descriptions that Birgit presented here.
Okay, thank you. And move on to the next question. So this is a question for Birgit, and the question is if you think whether these connectivity concepts could also be used in connection with diagnosis of neurological diseases, and if as among your long-term goals you would see a potential for connecting connectivity concepts to the diagnosis of neurological diseases.
Yeah, I think that is far too complex for what we are looking at right now. So mostly when we came up with these connectivity concepts, they were designed to make it easier to exchange theoretical models, which had relatively simple building blocks. Of course, you can also, in the meantime, just take actual connectivity data that people created in these large-scale projects and build this as a network. But to really compare then something you would define as a healthy network with a diseased network state, I think for that statistical analysis might also be useful, but on a whole different scale. So I don't think we can address that with these relatively simple connectivity concepts right now, unfortunately. We have to still learn more about what distinguishes healthy from pathological connectivity. What's an interesting question? Thank you.
Thank you. Then at the end of her talk, Birgit talked a bit about spatially structured networks, and here's a question for Mikael. Can you describe geometry-dependent connectivity in the connection set algebra?
Yes, you can. So allow me to be a little bit abstract first. So an alternative to regarding a connection set as a set of index pairs, you could represent it as a function, because for every possible connection that is a pair of indices, you could have an indicator function mapping that to Boolean value, telling whether that particular connection exists or does not exist. So in CSA, the concepts of functions on the set of connections is natural. And what you can do if you have functions on connections is that you can associate values to connections. And that could be common connection parameters like delay or connection weight, but it can also be a metric. So for a particular connection, you have a metric function which tells you how far away the source and target are. And if you have a metric function, you can use a mask that sort of cuts away, for example, connections outside a disk, similar to what Birgit was into. And you can also have probability operators that are dependent on distance. And of course, you're not limited to working with metrics. You can also have functions that actually map a neuron index to a position in a space. And this gives a big flexibility in CSA, such that you're not confined to one metric, but you could have multiple metrics, and you can have multiple metrics mapping indices to positions in a particular space. Mm-hmm. Yes.
Okay. Thank you, Mikael. And then there is a question for Sacha. So could you relate the connectivities we have discussed here to connectivity as one finds them in fMRI images, or let's say diffusion tensor imaging images?
The F in fMRI already says that this is functional connectivity that you obtain from such images. So in that sense, it differs from what we have studied here. So we have looked at structural connectivity, but that doesn't differ the principles. That doesn't change the principles according to which you could describe such connectivity or depict it. But in practice, the functional connectivity, it doesn't follow a particular high-level pattern exactly. So the connectivity that you would find that way would be more like an explicit list of connections that is covered by this X that I mentioned in the graphical notation. And the same basically holds for diffusion tensor imaging, DTI. Nevertheless, if someone created a model of such connectivity and then used concepts, high-level concepts, to approximate that connectivity, or maybe use high-level concepts and to compare with the observed connectivity, then again one could use what we have described, or some extension thereof that maybe takes into account clustered connectivity, as is often found in fMRI. So in fMRI you have these resting state networks, the clusters of areas that are highly correlated within each other, less correlated with others. And so to describe aspects of that, one could use high-level connectivity concepts.
Mm-hmm. And let me maybe follow up on this question a bit. Could you say, so, I mean, neuroanatomists have been looking at the brain for about a century now in great detail, collected a lot of information about what is connected with what in the brain. Has one used that to build models that incorporate the type of abstract rules that we've discussed here in the modeling process?
Well, thank you very much for asking me. Of course, yeah. So my group develops large-scale neural network models of cerebral cortex, especially of monkeys and human brains, that take into account a lot of details of the experimentally observed connectivity. For instance, from anatomical tracing studies, where a dye is injected into the cortex and then it is propagated via axons, either in the forward or backward direction, or for humans from DTI data that you mentioned before. And this connectivity is area specific, it's layer specific, and but nevertheless, below that level, the level of neural populations, within areas, within layers, we use random connectivity because it's not known well enough how the local connectivity is, especially at such a large scale and in individual brains. So it could also be a starting point for plastic networks that do become individualized and get more detailed microscopic connectivity. But yeah, the starting point could still be described with these high-level rules.
Okay, thank you. And then there is another question for Birgit that maybe follows up a bit in this direction as well. How would you address networks with more complex structures, such as layered networks or neuroanatomically inspired networks?
Yeah, I guess what one would do is to kind of define the steps in a maybe hierarchical way if you have a layered network. So define the concepts, connectivity concepts within the layer, between the layers, and essentially have a list or a graphical notation view of the network. But as long as the individual connection routines kind of can be conceptualized in that way that we described, it shouldn't be any different. So you just have a longer list of specifications, essentially.
Mm-hmm. Then we also have a very specific question here. So if you have the pairwise Bernoulli rule, should you have the same value of probability P for every region, or would that be varying?
That depends completely on what you want to model and what you think is appropriate. I mean, we all know that different regions have different connection probabilities, and also different neuron types will have different connection probabilities. So that is completely to the discretion of the modeler. I would say what you choose for your P or what exact concept you choose, but it doesn't have to be the same everywhere, of course. I mean, that's just a very simplistic connection rule for maybe very, very local networks where connection probability is approximately constant.
Mm-hmm. Okay, and then there is another question to Sacha, I think, following up on her previous answer. What is the connectivity pattern that you have seen most frequently in experimental models? Is that random connectivity pattern or more structured?
I'm not quite sure what is meant by experimental models, unfortunately.
Let's say, interpret that maybe as
published models.
Or let's specify in the kind of data that your group builds your models on.
Yeah, so the observations are always quite specific. So they give particular values that are they're never completely described by any particular high-level concept, but aspects of cortical connectivity that one may encounter are the distance dependence, so that's both local and between areas. The connection density decays approximately exponentially with distance. And the local connectivity appears quite random, but there are also there's an increased probability for neurons that share connections with a third neuron to also be connected to each other. There are often long-tailed distributions of numbers of connections, especially if you look at individual axonal and dendritic branches, that if there is at least one synapse between them, then there's likely to be more than one synapse. And so that sparse connections occur a lot, but some connections are very dense. And so those are some features that one gets. So one gets clustering of connections, both locally and also between areas. And there's a distance dependence and like-to-like connectivity. Neurons that are more similar also in their receptive fields or in their connectivity, they're more likely to also be connected to each other.
Thank you. Now we slowly need to round off, but there's one more, I think, very interesting question here. Maybe Johanna, you could comment on that. I have my own preferred way of creating network diagrams. Can I keep using it?
Yeah, so the idea of providing these standards that we have here in this study is that researchers reading the papers can directly know what it means without much further explanation. If you have and therefore we would for getting this intuitive understanding of the network from the network diagram. So if you already know the encoding of which element means what, therefore, in that sense, I think it is appreciated if the community evolves in this direction of using the same standards, but in particular, the standards that we provide here, they of course do not cover all networks that in particular more detailed networks and more detailed connectivity patterns, and therefore the notation that we provide is open for customization and for adding the features that one wants to represent. And in that sense, I would not, I would never try to stop you to using your notation. But if it's the best notation for the specific network that you have, but I would like to encourage you to explain in detail in the caption, in the main text next to the graphics, what the elements you use mean. And I think this is the most important aspect we wanted to convey with this graphical notation.
Thank you very much. I think that was a very nice roundup to the Q&A. We are unfortunately out of time, actually already 2 minutes over time. So I would like to thank all the speakers very much. Again, apologize to those of you whose questions I could not field now, and just add one little comment. There had been a question about application of network models towards diagnostic purposes. Other scientists in the Human Brain Project, in particular the group of Victor Girsa in Marseille with the Virtual Brain Project. If you Google TVB, you will find it. They are actually in clinical trials with network models at a much coarser scale that can be adapted to patient-specific models in the treatment of severe cases of epilepsy. So not at the individual neuron level that we have been considering here, but at the brain area model, there are models that are already on their way into clinical practice. With that, I would also like to thank all of you who joined us today to listen to our webinar and who came up with very interesting questions, made for an exciting question and answer session for coming. If you want to see a recording of this webinar or also the previous webinars, you can find them on the YouTube channel of the Human Brain Project. You see the link on the slide here. And if you have any other questions, you can contact us at outreach@Human Brain Project. And then I would like to welcome you all back for the next webinar, which will be on the 8th of March about landscapes of the human mind. And with that, I wish you all a good evening, a nice evening and a nice day, depending on where you are in the world. And thank you for attending.